Okay, next we are going to talk about cholesterol biosynthesis. So we discussed previously that liver will produce endogenous cholesterol in order to meet the body's demand. So how the cholesterol is going to be produced? So cholesterol is an essential molecule in the body. We know that, and it's an integral part of cellular membrane as well. And it's a non-fatty acid containing lipid, and it contains 27 carbon molecules. Okay, it will have this steroid ring structure and plus this aliphatic chain at the end, and totally 27 carbon molecules are present in there. So it maintains the membrane fluidity, and it acts as a precursor for several steroid hormones and vitamin D, and the synthesis of cholesterol will occur in the liver. So through the diet we are taking a limited amount of cholesterol, but the rest of the cholesterol will come from the liver. It's called endogenous cholesterol because it's produced within the body. So the synthesis is occurring within the liver, and there are 5 major steps involved in the cholesterol biosynthesis. It's such a long metabolic process, but please don't worry about all these pathways or all these steps. So I'm going to talk through the important steps, and you need to remember those important steps and the important enzymes involved in these pathways. So what are these 5 major steps here? So the first step is called mevalonate synthesis. So again, cholesterol biosynthesis will utilize acetyl-CoA, the 2 carbon containing molecule. So using the acetyl-CoA, it's going to produce a molecule called mevalonate. That's the first step. Second step is called isopentenyl pyrophosphate synthesis. Okay, it's called IPP, isophentyl pyrophosphate synthesis. Third step is called squalene synthesis. Fourth step is called steroid nucleus formation, because that's when the steroid nucleus is going to be formed. And then the fifth step is cholesterol formation from lanosterol. So these are the important steps that we are going to talk about in the cholesterol biosynthesis. So first step, mevalonate synthesis. It's an important molecule during the cholesterol biosynthesis. So how this is going to be produced? So we need 3 acetyl-CoA molecules in order to produce mevalonate. Okay, so acetyl-CoA, as we know, 2 carbon containing molecules. So first, two acetyl-CoA molecules joining together in order to produce 4 carbon containing molecule called acetyl-CoA. Okay, so this is happening. The thiolase enzyme is basically combining these two acetyl-CoA, liberating one coenzyme A, and then producing 4 carbon containing molecule called acetyl-CoA. Sorry. Then the third acetyl-CoA is being added using an enzyme called HMG-CoA synthase. Okay, so the third acetyl-CoA is being added to these molecules. So finally, you will get a 6 carbon containing molecule that's called beta hydroxy beta methyl glutaryl CoA. So this is also liberating a CoA. Okay, so now there's an isoenzyme of HMG-CoA synthase in mitochondria that's involved in ketone body synthesis. Don't get confused between these enzymes. Yeah, HMG-CoA synthase here is producing HMG-CoA, but in mitochondria it will produce ketone bodies. Now this HMG-CoA, that's beta hydroxy beta methyl glutaryl CoA, will enter into endoplasmic reticulum. Okay, moves to endoplasmic reticulum from cytoplasm, because this is happening in the cytoplasm. Now this molecule will enter into endoplasmic reticulum. Here the mevalonate is synthesized by reduction by using an enzyme called HMG-CoA reductase. Okay, so you produce this HMG-CoA. HMG-CoA is coming into endoplasmic reticulum. Here there is an important enzyme called HMG-CoA reductase that's basically reducing this molecule, this HMG-CoA, to produce mevalonate. So here it's releasing to an ADP and coenzyme A. So finally you are getting mevalonate, which is free from any more coenzyme A. Okay, so this HMG-CoA reductase is an important enzyme because that's a rate limiting enzyme for cholesterol biosynthesis. So you must remember the name of this enzyme, HMG-CoA reductase. It's a rate limiting enzyme for cholesterol biosynthesis, and this is the enzyme target for statins. So we take statins during cardiovascular disease progression in order to control our cholesterol level. So the statin will bind to this enzyme and block the cholesterol biosynthesis or reduce the cholesterol biosynthesis. Okay, so mevalonate a 6 carbon containing molecule. It's being produced utilizing 3 acetyl-CoA. The important enzyme here is Kachamji-CoA reductase. That's a rate limiting enzyme controlling the entire cholesterol biosynthesis. And that enzyme is the target for statins to control cholesterol biosynthesis. Now, the next step is IPP. Okay, IPP synthesis. IPP means it's basically isopentenyl pyrophosphate. So using mevalonate, IPP is going to be produced. Okay, again, there are a couple of steps here. First, the mevalonate kinase enzyme is coming, and that's adding phosphate molecules to this kinase. Okay, so first it's adding one phosphate group and then another phosphate group, then it's producing pyrophosphate mevalonate. So this pyrophosphate mevalonate then releasing carbon dioxide, okay, utilizing energy, and then finally producing isopentenyl pyrophosphate. Okay, so here the decarboxylase enzyme is removing a carbon dioxide. Therefore, it's producing this IPP molecule, which is 5 carbon containing molecules. Some of these IPPs will be converted into its isomer that's called dimethyl allyl pyrophosphate. So some IPP will stay intact as IPP. Some of this IPP will be converted into its isomer by using an enzyme called IPP isomerase. It will be DMAP, yeah, DMA APP. So now these two molecules are ready and each molecule contains 5 carbon. Remember, okay, even though mevalonate contains 6 carbon, what carbon dioxide was released, therefore each will have a 5 carbon here. Now the next step in cholesterol biosynthesis is squalene synthesis. That's the next molecule is going to be produced. Okay, squalene. So here both IPP and the DMAP will be utilized. Okay, in the first step, it's a condensation step. So basically one IPP and one DMAP is going to be joining together through head-tail condensation. Okay, so I labeled all these things properly here. This is DMAP, this is head portion, this is tail portion, and this is IPP, this is tail portion, and this is head portion. So basically the pyrophosphate containing end will be head portion, the other end will be tail portion. So there's a condensation process happening here. This is basically head to tail condensation. So this position is going to join here in order to produce the next molecule. Okay, so totally next molecule will contain geranyl pyrophosphate that will contain 10 carbon because this is 5 carbon, this is 5 carbon. It's joining together by removing pyrophosphate and then producing 10 carbon containing molecule called geranyl pyrophosphate. Now there's an addition of another IPP. Okay, so here again this geranyl pyrophosphate head to tail condensation with IPP, another IPP, then it's producing the next molecule called farnesyl pyrophosphate or FPP. Okay, so finally 2 FPP molecules because each one containing 15 carbon now. Okay, remember this FPP will contain 15 carbon because 5 here and another 5 here. So totally 15 carbon molecules. So finally it's producing squalene, which that's basically head to head condensation. So basically here this is the head portion and another molecule FPP will come. The head and head will join and that will completely release all the pyrophosphates and then it will produce a 30 carbon containing squalene molecule. Okay, I hope you guys understand this and following me. If you don't understand, please let me know. So we are starting with the 5 carbon containing molecules, IPP and DMAP. So a DMAP is basically isomer of IPP. Exactly same molecule, just this double bond position will be different. Okay, now head to tail condensation is happening first. So 2 of these molecules joining IPP and DMAP are joining together to produce GPP. So that's a 10 carbon containing molecule. Now the GPP is condensing with another IPP, head to tail condensation, producing a 15 carbon containing molecule that's called FPP. Now 2 FPP molecules joining through head to head condensation, removing all the pyrophosphates completely from the structure. Okay, that's why it needs head to head condensation. Now it's producing squalene. Squalene is 30 carbon containing molecule. Okay, so this is the reduction reaction. So it requires NADPH. Okay, it's utilizing the energy here. So this step removes, the final step will remove the pyrophosphate. Now the steroid nucleus is going to be formed. So we got 30 carbon containing beautiful squalene molecule. Okay, but now we need to make the steroid structure. So the steroid nucleus is going to be formed here first. So basically it's addition of a single oxygen molecule will produce squalene epoxide. Okay, this is an enzyme called squalene monooxygenase enzyme. This will add an oxygen to squalene and release water molecule and it will produce squalene epoxide. Okay, so these molecules again being converted into other molecules through an enzyme called lanosterol synthase. So lanosterol synthase will add hydrogen molecules and forms an intermediate that's called protosterol cation, and then subsequently it will produce lanosterol by removing the hydrogen. So initially it's adding the hydrogen and then later on it's removing the hydrogen and finally it's producing a molecule called lanosterol. So here you need to remember these two enzyme names, squalene monooxygenase. That's the one adding the oxygen molecule to the squalene, and by adding this oxygen molecule, it's starting to make the steroid nucleus. Then the next important enzyme is lanosterol synthase. That's what is filling all these gaps in the nucleus structure and making a stable lanosterol molecule in the end. Initially, it's adding a hydrogen and then removing a hydrogen. OK, so now we have lanosterol, which is containing basically steroid nucleus that's necessary for cholesterol. So the last step here is the cholesterol formation from lanosterol. So by utilizing the lanosterol, now we need to produce cholesterol. So here 19 steps are involved. OK, we're not going to go through in detail of all these steps. OK, and it's excuse me, and it's not important for you guys as well. So you don't have to remember all those 19 steps. So basically what's happening here is from lanosterol, one formic acid is being removed and then two carbon dioxide molecules are being removed. So formic acid containing one carbon and obviously carbon dioxide will contain one carbon. So totally one formic acid and two carbon dioxide will remove three carbons. So squalene, as we know, it contains 30 carbon. Lanosterol will have the same 30 carbons. Now by removing three carbons, we are producing 27 carbon molecules containing, 27 carbon containing cholesterol in the end. OK, so that's what is happening here. There is 19 steps this lanosterol has to go through. But the main point here is it's removing one formic acid, two carbon dioxide molecule, and then finally cholesterol will be produced, which will contain 27 carbons. OK, so we started with two carbon containing acetyl CoA. Now we have 27 carbon containing cholesterol molecule. So that's about cholesterol biosynthesis. You need to remember the important steps, important enzyme names, particularly HMG-CoA reductase and the lanosterol synthase and monooxidase for squalene. And then the important byproducts we are getting from end of each step, mevalonate, HMG-CoA, and then here squalene, lanosterol, and these type of molecules, IPP, DMAP. So that's what you need to remember. Don't worry about the structures of this molecule, but you need to remember how many carbons are there in each of these molecules. OK. OK, now we are going to fate of cholesterol molecules. So cholesterol synthesis is finished. We learned about how the acetyl CoA is being converted into cholesterol in the end. Now, how cholesterol is acting as a precursor to produce various molecules. So cholesterol is synthesized in the liver, as we know, and it's being transported through LDL and it's going to all the other cells. Then it's going to be utilized as a precursor to synthesize steroid hormone, bile acids, and vitamin D. So I'm not going to go through in too much detail about all these processes, but the summary is here in this slide, which you can go through. So cholesterol is being converted into oxysterols. Again, that will lead into steroid hormones, and you can see all different types of steroid hormones produced using cholesterol. For example, progesterone, cortisol, corticosterone, aldosterone, testosterone, estrogen, all the enzymes are, all the hormones are being produced by utilizing cholesterol as a precursor. Then bile acid being produced by utilizing cholesterol, and some of these bile acids will be excreted as well. And the bile salts are also produced as well. Okay, so most of the steroid hormones, bile acids, and vitamin D are being produced by utilizing cholesterol as a precursor. Most of the cholesterol will be recycled in the body and some will be excreted mainly through bile. Excess cholesterol will prevent the synthesis because it's a backward negative negative feedback regulation. So it will be stopped as well. So remember this, cholesterol is being used as a precursor to produce bile acid, and then it's used for corticosteroid production, estrogen production, and vitamin D production as well. Okay, so please go through all of this and then let me know if you need any further details. And as we discussed right at the beginning in the last lecture, excess amount of cholesterol will lead to atherosclerosis. Again, you will study atherosclerosis, hyperlipidemia, all the cardiovascular diseases next year, so you don't have to worry about them this year. So there are key steps here for cholesterol metabolism. In summary, cholesterol is being synthesized in the liver. Cholesterol synthesis involves 26 steps in total, and it occurs in 5 different stages. Okay, and HMG-CoA reductase is a rate limiting enzyme here that controls cholesterol biosynthesis. And the cholesterol is used as a precursor for steroid hormone synthesis, bile acids, and vitamin D. And most of the cholesterol is recycled and a small amount gets excreted through bile in the gut. And excess cholesterol prevents the synthesis, and the excess cholesterol will also lead to atherosclerosis. And it's extremely important to control the cholesterol level in order to prevent the progression of cardiovascular diseases in the body. Okay, and the statins, that's a cholesterol reducing drugs, that's mainly targeting HMG-CoA reductase to lower the cholesterol levels in the body. Okay, so you can also go through some textbooks. Any biochemistry textbooks will talk about cholesterol metabolism and fatty acid metabolism. So for example, the biochemistry by White and White, that's a really good book, or you can study Stryer or Leninger as well. Okay, and if you have any other doubts, please let me know and I'm happy to go through this with you again. So thank you. This is the end of this lecture and I will see you guys in the next lecture in next year or during tutorial session this year. Thank you guys.